This is TWIS. This Week in Science, episode number 789, recorded on Wednesday, September 2nd, 2020. Science with a twist. I'm Dr. Kiki, and tonight we will fill your head with holes, mergers, and something special. But first, thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. Earth. It's at most a medium-sized planet with a lot of humans. Billions of them even. More humans than you are likely to run into on any other planet. And despite sharing the same star, each one sees the world in a different light. For the most part, we humans walk past each other, seldom knowing how or what the other thinks about this or that. But every once in a while, we cross paths with one of these humans and strike up a conversation. Every once in a long while, we find that there's another human who sees the world very much as we do, has a similar ethic or aesthetic appreciation, chases similar sparks of curiosity into the weeds of contemplation, lighting up conversation's path like lightning storm on a dry summer's night. Ed Dyer, longtime listener, contributor, and conversation starter here on TWIS, was one of those humans for me and for a lot of our TWIS family. His spark will be missed but not forgotten, as he has inspired many of us to keep setting small fires to share our thoughts around and enlighten our paths as we walk this world together. Thank you, Ed, for being a part of why we do This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're back. We are back. 3 voices to talk about science for a night. And we are missing our dear friend, Ed. For those of you who knew him in the chat room, there is sad news of his passing has come to light. So he's missing from our chat room tonight, and he's going to be missing from our lives. But his spark will always live on. Thank you for that great disclaimer, Justin. But we have a great show ahead. And in Ed's honor, we are going to hopefully light a spark, hopefully drive some curiosity. I think Ed would want us to keep doing that in the way that we do.
Absolutely.
So on we go with the show. What do we have lined up for the show tonight? I have stories about some of those mergers and holes, black holes to be precise. I also have stories about brains because brains are fascinating to me, and yeah, that's where I'm going to be heading. What are you doing, Justin? What are you going to talk about?
I've got a gut microbiome story. I have got how insects could save the planet, as well as some insane things that bacteria can do that I didn't know that they could do, that maybe nobody knew that they could do, but they're doing it, and so they can do it.
I want to know what they're doing. What are they doing?
Yeah.
You can wait for the show, Kiki.
Yeah, it's a teaser. This is just to keep you watching. Oh, and a not a COVID cure, but a last-ditch thing that reduces fatalities. That based on, well, that's the whole story, basically.
Let's talk about that in our COVID update.
A little bit of sort of silver lining in a dark cloud.
I hope so. Yeah. Blair, what is in the animal corner?
That's a great question. I plumb forgot. No, I have a bunch of information about taking better care of lab mice. I have anemones and a little bit about women in science.
Sounds like great news. Can't wait to hear it. So let's dive into the show. And for all of you, if you have not yet signed up for This Week in Science as a podcast, we are available as a podcast pretty much everywhere you find podcasts. Look for This Week in Science. It's that easy. We're also available on YouTube and Facebook, and we now are on Twitch. So try and find us there under TWIS, TWIS Science. TWIS Science. Only one S in there.
This weekend,
and you can
science.
This week in science. Yes, science. And our website, twis.org. Find us if you can. All right, into some quick stories to start our show. I have a story. Let's start it off with that black hole merger. Big story out this week that we actually already talked about back in June. Wait, what? Yes, LIGO Virgo. We do love LIGO Virgo. And the LIGO Virgo collaboration, which looks for gravitational waves, back in May, detected 4 little blips in space-time. Little tiny 4 little blips. Like they basically turned on their device and it was like, oh, what are those 4 little blips? But it took, much cleaning of the information of all of the multiple blips to be able to even detect these almost nearly undetectable blips, which they have now confirmed. Pretty much, they've run analyses and come to the conclusion that the most likely explanation for what they saw in these 4 little blips was the merger of 2 stellar mass black holes into a larger intermediate-sized black hole. And this is very exciting because we have evidence of stellar mass black holes, and we have lots of evidence of the big supermassive black holes, but there's really no evidence of the formation of these mid-sized black holes. And if theories are correct about black hole formation, that as you put black holes together, smaller ones will make bigger ones, and those bigger ones will make even bigger ones. If that's correct, then they should have seen this, but they hadn't before. So most likely explanation is that this is the merger of 2 stellar mass black holes to create an intermediate mass black hole merger, which has never before been seen. Something new! Go science. Did you have another story about a merger, Justin?
This is that thing that we were talking about already at the beginning there. Like humans often do, bacteria will live together in communities. They're working together, taking on different tasks, individual bacterial tasks to sort of fill out the needs of the society, as it were, of the bacteria. So understanding how these interactions work is very important to how antibiotics work and how infectious disease happens and this sort of thing. Researchers at the University of Delaware, while doing that sort of research, discovered that bacteria can do more than just work together. They found bacterial cells from 2 different species, although closely related, can combine into a unique hybrid cell by fusing their cell walls and membranes and sharing the cellular contents of the internal portion of the bacteria, including proteins, ribonucleic acid molecules, which regulate gene expression, control cell metabolism. In other words, the organisms exchange material and lose part of their own identifiable identity.
They lose their sense of self.
Is like, I assume there would be 2 nuclei, but...
So yeah, this is going way beyond like gene transfer. This isn't little proboscis things transmitting, spitting DNA into one another. This is apparently an unprecedented observation, which has apparently not been seen before, which is why it is unprecedented. That's the meaning of the word. So this was reported Tuesday, September 1st, in Bio, which is a journal of the American Society for Microbiology. And they think this could shed light on some unexplained phenomena affecting human health. This could be applied to energy research, biotechnology, a whole lot of other things. Yeah, so they mix that. This is a quotey voice from, oh boy, this is going to be a fun. Eleftherios, it's a Greek name. This is the research team is led by Eleftherios Papotakis. That's probably pretty close. Who's studying the interactions between Clostridium ileum dallii and Actobutylicum. These are two Clostridium bacteria that are in the same clade, but they're different species. And this is the quote from Papotakis. They mix their machinery to survive or do metabolism. And that's kind of extraordinary because we always assume that each and every organism has its own independent identity and machinery. Previously, researchers had observed that bacteria could exchange some material through nanotubes. This is that horizontal gene transfer. But the combination into turning into a hybrid cell itself was unexpected and unseen. This is Camille Charrubin, who is a doctoral student in chemical and biomolecular engineering and first author of the paper. This is the first time we've shown this in bacteria, and it's also a new mechanism for how material is exchanged between cells, which is because they became the same cell. So why would they do this? Apparently, this is what's really they are sharing by sharing this machinery. They're also creating things that the other needs to exist. One bacteria is able to create a protein that the other might not be, and they both get to use it. And it just lessens the amount of work that each needs to do and gives them advantages and access to things that they may not be producing themselves. Pretty amazing. Really. This is like new. This is like, if like two humans could do this. This isn't just like information sharing or flapping a gene. It's like, hey, let's be conjoined twins. You wanna do that? Like, forget getting married and like sharing a bank account. This is like, you know what? My liver's not as good as your liver. Why don't we just scooch together? We'll use your liver. My heart's really good. We could just use that one. That's all we need. It's really wild. And it makes me wonder, the extrapolation that immediately jumps to mind into this is like, there's so much speciation and evolution that takes place, but this is like, could these possibly reproduce in this state? And then what does that look like? I mean, because they're doing this without sexual genes involved.
Right. It's not sexual reproduction, but then, I mean, what this really gets at for me is that there is, for bacteria at least, there is very little fine line between different species. That, the fact that they can merge so easily, that there's blending that can take place, that, we already knew about horizontal gene transfer. This is just like a ramped up version of that, where they're like, let's just work together here and become one. And these hybrid species could then have, if they work in that environment, then they work well. If they don't, they die out. That natural selection would really act on them in that case. I mean,
from an evolutionary point of view, though,
what is to keep the membranes from? These are just lipid bilayers, right? What is to keep lipid bilayers in a situation from melding and blurping together with other lipid bilayers?
I guess if you just don't do it right, both cells might die, or one might.
Yeah.
But this is evolutionarily, is this like the half step to the first mitochondria? I mean, is this sort of like...
It sounds like it would be.
Yeah. So we know that bacteria can just sort of do this occasionally, I guess, at least. Then that pathway for our evolution looks like a lot more possible. Like, we've seen the halfway. We have the missing link to merging bacteria. You just might get to a point where one's like, yeah, you can come in, but I'm not really going to share everything.
Yeah.
I'm just too big, maybe.
Hey, babe, you want a smush? It might kill us, or it could make
talk
us
about
way better.
mashing. Talk about mashing. This is the real deal. Anyway, pretty fascinating.
I think I would love to know more details on, where and when this has a tendency to happen, or where and when it does not. So when, if this is one of these early steps to things like mitochondria coming in, being, and like, I guess, eaten and not endocytized, but just brought into another cell. How does that happen? How does one smaller bacteria get brought into another bacteria and kind of consumed in that way, but not destroyed? How does that end up? Why? And
if they're sharing, if they're at the point where they're sharing, ribosome and RNA, that's just a hop, skip, and a transposon away from sort of gene transfer within the cell, which is how you could get like that. This is in the prototype eukaryote, where you could sort of see like, oh, this is how it's possibly translating not just the bacteria that's sexually reproducing on the exterior cell, but it's also bringing in those elements that are in that cell that it's absorbed. Because with time they can actually start to mix in that. And then, anyway.
It's interesting.
Yeah.
Very thought-provoking. Thank you.
Yeah.
Blair, did you have something special to talk about?
Yes. Female scientists, they're important. This is a study from the University of Maryland, Baltimore County, and this started out as a birdsong study. Even just a couple weeks ago, maybe even last week.
So tweet.
I read it. I talked about a story about bird songs, and even said in the story, as it was listed, the majority of birdsong is carried out by males to woo females. But it actually turns out that maybe that's all sexist BS. So basically, it turns out from a 2014 study, a PhD in the biological sciences of a woman found out that about 70% of female birds sing. So already that's kind of thrown out the window. But also that both sexes almost certainly sang in the common ancestor of all bird species. So this kind of rocked ornithology at the time. But since then, the researcher who did that, Karen Odom, and teamed up with Casey Haynes, who's a PhD candidate from 2019, who is a biological sciences PhD candidate. They worked together to document what has been going on in this field with researchers. So women are more likely than men to be authors, and even more likely to be first authors on papers about female birdsong. So women specifically have reshaped this field of study. So this again reminds us that a diverse group of researchers is critical for scientific innovation. And diversity could also help, as we've discussed before, a more accurate and complete understanding of biology, of science, of everything. So this is something that completely got rocked by a few female scientists and then just kept being found again. So these male scientists, it appears, were carrying a bias, assuming that males were singing for females. But once you kind of peeked in over the under the covers, I guess, of the bird song situation,
under the hood,
it's under the hood, it's under the beak. It's very different. So there's obviously a bias there. There's also an interest, people research what they can relate to. And so perhaps this was also something that female researchers were interested in, because male birds were historically researched before that because they were more interesting, because they're prettier, because they sing more.
They got the plumage, and of course, but you think they're singing more.
Right.
Yeah.
And so it's just a reminder that representation is key in the sciences, and that we need many voices in a lab doing research, because there's an implicit bias no matter who you are, no matter what you're doing. And so if researchers all look exactly the same and have the same background and have the same upbringing and all this kind of stuff, it can impact research.
Yeah, it can also impact the way that research is done because coming from a female-run bird lab, we weren't studying song, but I was in the same building with one of the fathers of birdsong. And his lab, he had, at the time I was there, he had a female postdoc who was working with him. And but everyone talked about the lineage, and I love science for the way that people track back their lineage through from grad student to professor and back through time. And so it was, this individual, and then talking about his mentor, the PI that he worked under, who was another man, and it goes back and goes back, and if the people in charge of the labs who are running the labs are thinking about things in a particular way, even if they have women or, a diverse representation, they're not, the people who run the lab get to tell the people who work in the lab kind of what questions get to be asked, is what I want to say. So those perspectives are important. And so the lineage may start somewhere with someone's idea, but through the generations of scientists, there can be evolution.
Yes,
and so,
absolutely. Yeah.
Yeah.
I mean, it's a lot harder. Also,
saying.
if you're just talking about general diversity, it's a lot harder to increase diversity in a workforce if your entire team of VPs are all white men. So yes, it's definitely, it affects things, and
Yes.
And it affects the way people look at the world, whether we want to think that we're 100% impartial or not.
And there's something to the whole learning something very complicated. We're dealing with a large set of information. You start to need to make some assumptions. You don't go in as an engineer and challenge every law of physics as you go along to get to the point where you can comprehend how to build a bridge or whatever. You make assumptions that these things you're being taught are pretty set in stone as you go along and fill in that information. So that's also part of why it's then hard to break out of that, because where do you start challenging knowledge that has been taught and is assumed to have been well tried and tested? The thing, though, that this whole conversation made me think of is, the common ancestors to all birds probably sang, since pretty much all birds sung or sing. Does that mean dinosaurs?
All dinosaurs sang.
All singing all the time? Like T-Rex, as big and ferocious as a scavenger type predator as it may have been, had the most lovely lilting song every morning.
Do you know the sound
T-Rexes
make?
The T-Rexes thought they had a pretty song, but because they were very big animals, it was more of like a blah.
But all the other dinosaurs were like, "Oh, that's pretty. We're clapping because you can't." But that's good. It's a great song.
Yeah, good job there.
Yeah, I mean, crocodiles make very silly sounds for the giant, scary creatures that they are.
Oh yeah.
I think now I want to know why female birds would sing and why male birds would sing, and this, if female birds are singing to get the attention of males, which species do this? And I mean, I think we're talking about this, but still, if you go out in nature, you're not going to be seeing female birds out on the tree limbs doing all of the singing. So in passerines, I want to know really what the split of birds who are doing this is.
Right.
I'm not saying I don't believe it, but at the same time, I'm like, I believe it, but I'm going to guess it's in certain situations where it has evolved in a very particular way, where, say, for instance, the Western scrub jay does not have a lot of distinction between the male and the female body forms. So, and they share in helping at the nest. And there's all sorts of things that are very similar. So why wouldn't they both sing?
Yeah. If I may, we just talked about flamboyant cuttlefish last week. The females also can make brilliant displays. They're not trying to woo anyone, but they use them to communicate with the males who are wooing them.
Yeah.
Mm-hmm.
Why not have a conversation?
Yeah, absolutely.
I think you're pretty. Well, I think you're pretty.
I will accept this praise. You may come closer and I won't try to kill you.
I'm not a black widow. Okay. Yeah. All right. Well, this is a fascinating topic I want to learn more about. How about another topic that is really interesting? Water. Water. Where does it come from? Where does water come from? Where have we discussed water on Earth coming from?
Comets.
Yeah.
From space.
Or maybe not.
Or
maybe not.
What?
Yeah.
What? Huh? No,
yes.
It's from space, isn't it? Oh.
This is what we think because we look at meteorites. We look at these little chunks of rock from space and look at how much water's in them and get an idea of like, okay, where could water have come from? And the reason that they thought it came from outside of the inner solar system, out past Jupiter, is because that's kind of out where things get frosty and also where water could have stayed out there in ice form, been able to travel long distances, and then landed on our planet after it was heated up. And, our planet went through a lot of transformation over the years. It got hot. It might have lost a lot of water. The likelihood that it started with water was really not considered because they were looking at these different kinds of meteorites, the ones from outer part of the solar system. Yes, they had a lot of water in them. And these chondrite meteorites were being looked at, and they were finding those that had makeup very similar to the Earth's. So it's like, okay, this stuff seems like the Earth, so it could have made up the Earth. And they thought these carbonaceous chondrites were the ones that carried water to us. However, they don't really have the same isotopic or molecular makeup as Earth. But they were from outside the snow line and could have held water. So, hey, let's think about those. But some researchers just published in Science about some new analyses of enstatite chondrites that they have looked at. These enstatite chondrites were formed in the inner solar system in the rocky area. And nobody was looking at them before because they were like, they're all rocky. They can't hold enough water to cover the Earth in water. Well, apparently, looking at the water concentration in these enstatite chondrites and the deuterium to hydrogen ratios within these little meteorites, they match the Earth's interior. They match the rocks of Earth. And so what they are proposing is an alternative hypothesis that the Earth could have formed from these enstatite chondrite meteorites, that they could have had enough water to cover the whole planet. The only issue is maybe if the enstatite chondrites, maybe if there was vaporization, like I said, during the heating part of Earth's formation, then vaporization would have lost water. But then maybe it was brought back by those the carbonaceous chondrites later. Maybe comets brought some of it, but not all of it. And that's what the scientists in Science propose. They said, why do you have to make up some complicated story like Jupiter had to move into the inner solar system and get all pushy and move meteorites out of orbit and move comets out of orbit and then move back out, and then that would lead to all this? Why do you have to make such a complex hypothesis? They said, this is much simpler. Earth formed from the rocks where it lives.
Yeah.
In some ways, it's simpler.
Yeah. Makes more sense.
Yeah.
In some ways, it's more complicated.
Yeah.
I don't know. Yes, you could have made the cake from the ingredients in your cupboard, but did you see that cake? Because it would have been a lot easier to just buy it at the store.
It could have been easier to get it from the store, right? So these are now dueling hypotheses, but there is evidence now from actually looking at these meteorites that maybe it isn't a cut and dried kind of explanation of water from the outer solar system riding on comets.
There's nothing cut and dry about water.
Ha 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 ha. Dun, dun, dun. All right, Justin, how are insects the future? What's
Oh,
what?
we are really moving along. All right.
Yeah.
Well, let's see. Where's that? Oh, yeah. So this is one of the ways in which global warming can be combated. Might come from this unlikely source: insects. Turns out we could simply breed more of them, a lot more of them, enough so that they could block out the sun.
What?
No.
No.
Or we could or we could just use them for food. So agriculture is currently practiced. The current agricultural practices that we use are one of the major drivers of carbon emissions. This is out of Indiana University / Purdue University, Indianapolis, which is a university proving that you need not settle on a single name before you combine them, like hyphenated university. Scientists have found new evidence that previously overlooked insect shows great promise is an alternative protein source. It is the yellow mealworm, which doesn't sound like too far off of things that we've seen before.
We ate those in Philadelphia. However long
we ate
ago that was.
We didn't eat the yellow mealworm. So this
We
is
did?
no, this is a different mealworm, a different worm. But this one specifically, research is based on new analysis of the genome of the yellow mealworm species, Tenebrio molitor. And this project was led by Christine Picard, Associate Professor of Biology and Director in Forensic and Investigative Science. This is a program where she's no doubt asked daily to say, "Make it so." The work was published in the journal Insects as Food and Feed, which is a very specific journal. It's pretty much dedicated to eating bugs. This is quotey voice from Picard. Human populations are continuing to increase, and the stress on protein production is increasing at an unsustainable rate, not even considering climate change. The research was conducted in partnership with the Beta Hatch Inc. Has found the yellow mealworm, just traditionally a pest bug, it's crop eater, can provide benefit in a wide range of agricultural applications. Not only can it be used as an alternative source of protein for animals, but its waste is also ideal as an organic fertilizer. Picard and her team sequenced the yellow mealworm's genome. Results will now help those who wish to engage the DNA to optimize the yellow mealworm for mass production and consumption. Insect genomes are challenging, quotey voice from Picard, and the longer sequence of DNA you can generate, the better genome you can assemble. So some of the uses, mealworms being insects are a part of the natural diet of many organisms.
They can be yummy, very nutty flavored.
Yeah.
I've had them I've had them in cookies.
So these are the mealworms. I looked it up. They're the darkling beetle mealworms. They are the ones that we ate in Philadelphia.
These are?
Mhm.
Oh.
Yeah,
We
ate these.
Yeah,
Great.
so they're commonly roasted, but they're even more commonly crushed into flour. They make a really kind of nutty flavored, but really protein rich flour that you can use in pretty much anything that needs flour.
So one of the things that she's sort of indicating is that we should really, we could actually use these to feed the food that we eat. So fish. So if you have fisheries, you could be feeding them to the fish. Pet food industry can use them as a protein source. Chickens. You can feed chickens with these mealworms.
Oh yeah, we feed mealworms to chickens at the zoo.
Yeah, perfect.
That makes perfect sense.
Wait, why
I
do you
fed
have
mealworms to chickens when I was a
In
kid.
the family farm.
What kind of side of the road?
No. It's part of the family farm.
We got a cyclops pig. Pig with one eye. Lost it. That's all. Not really a cyclops. Just a pig with one eye. Yeah, and again, humans again also. I didn't realize this is the same one because this sounded like it was like the first time they discovered that we should be eating this. But I guess what this is saying is based on the genome, it's an ideal source of protein.
I love the words, though. We will optimize the mealworm for consumption. It's going to become No, it's going to become like the next cow. It's going to become the next chicken. It's because we have optimized those animals not through genetic modification of the genome, but through selective breeding, which was modification
which is
modification of the genome through breeding. But now we have, the big breasted chickens and, cattle that either produce massive amounts of beef or lots of milk. They are very specialized for what they do as food creatures. And so I find it very interesting that we're starting to look at insects in this same capacity.
So not to like do a product placement. That's not my intention here. But I also have eaten on many occasions something called chirps chips, which are made out of crickets. And they taste great.
I don't like crickets, but that's okay.
So what's also interesting, though, I think, is that you even when you have the genome, sometimes the best path forward is still breeding. The biotech industry uses this a lot, where they have a couple of different strains of yeast or an E. coli or something. And they want to get these certain sort of robust gene combinations out of the different disparate ones. It's actually sometimes more difficult to splice them in and see what downstream effects you get than to take them and start breeding them in these massive breeding programs.
Why? Sorry, go ahead. I was interrupting with my thoughts.
Oh, interrupt away. This is the whole point.
Why have none of these insect companies started making an insect burger to compete with the lab-grown meat?
That's a really good question.
You have your steak burger, your turkey burger, your mealworm and cricket burger, why aren't we doing that?
I will say the one weird thing about eating mealworms or crickets is that if you have a shellfish allergy, it is likely to be an issue.
That would be the stop.
That is something that
is that with crickets as well, or is that just the
it's because exoskeletons have the same kind of proteins in them as the exoskeletons in shrimp and crab.
That's why it's not in hamburgers.
It's not going to be in hamburgers, but it could. Why? But there still could be insect burgers.
Yes.
With a big disclaimer.
Put it on the seafood aisle.
Yes.
Confuse everyone.
Do you see the danger? One more for the quick stories. I need to tell you about how the roads kill. That's right.
Via drivers in cars?
Nope, not via drivers in cars. The roads themselves.
Because it's hot?
Because when it's hot, they heat up and they release pollutants into the air. They release organic compounds and other things called secondary organic aerosols, major contributors of what are called PM 2.5 air pollutants comprised of particles that are smaller than 2.5 microns in diameter, which are pretty bad when they get into your lungs and can have significant effects on public health. Researchers published in Science Advances, these researchers are from Yale University in the lab of Drew Gentner. They looked at roads, asphalt heated to different temperatures, and determined that these products emit substantial pollutants into the atmosphere without any cars involved. It's just the heating. Paved surfaces and roofs make up about 45% and 20% of surfaces in the United States in cities, respectively. These surfaces saw a jump in emissions up to 300% for road asphalt when it was exposed to moderate solar radiation. So heating plus solar radiation is even worse. So we should maybe start looking at different ways to create asphalt, because asphalt is not helping us any. Well, it is. It helps us get around. It's great if we can make it nicely and neatly. But
there is one thing that we can do without getting rid of cars right away, which is greening roofs. So that was one of the things that you were just talking about.
45% of cities have asphalt.
they contribute to pollution like this, but they also contribute to urban heat islands. So
Yeah.
because they heat up and release heat, it impacts the whole microclimate of an urban area. So greening roofs, planting on rooftops can have a huge impact on both of those things.
Can I just say, I find humans to be very inefficient when it comes to roofs.
Yes, agreed.
Three things, especially today's
more
day and age.
more things.
One, you're absolutely right. All roofs should be painted white to reflect heat in the warm areas. Yeah, absolutely.
We're covered in greenery.
Two, they should also then have solar panels.
Yeah.
3, forget both of those. Why doesn't everybody have a deck on their roof that they can enjoy with a little bit of a view?
Mm-hmm.
How come that's so rare? It's like the most obvious thing. You could put the deck on top of the... Anyway, people are just inefficient and uncreative when it comes to their roof. It's like an oversight.
Or people have inherited or purchased buildings that have been made in one way for a long time. And we need to change it.
Yep.
Yeah, it's that, but I think it's also because it's unsafe. I think it's like when you see somebody whose hair looks great and then they walk by and the back is all messed up. It's because they just didn't see it in the mirror.
I feel like I'm being attacked right now.
Oh no, I'm so sorry. No, that was in general. That applies to all humans.
I know, that's like anybody with curly hair at all. You like get the front all figured out, and the back is just a frizzy just bump.
You don't know, and you don't care, and nobody's going to tell you.
Well, especially now where you're on camera all day, every day. No one's looking at the back of your head anyway. It's fine.
Nobody's looking at anything. Oh, this is This Week in Science. Go take a look at your roof. I hope that it's a nice roof. If you just tuned in, I hope that you are enjoying the show. We have a lot of science to come. If you are interested in a TWIS shirt or in a mug or in some kind of TWIS merchandise, head over to TWIS. TWIS, what's my website? Yes, TWIS.org, TWS.TW. I can't speak. Head over to TWIS, TWIS.org. And click on that Zazzle link to see what we have on offer and support the show. Okay, are you ready for a COVID update? Yes. All right. Let's talk about COVID-19. I don't have a huge COVID update. I know Justin has a story coming up here. The United States is starting to come out of the second resurgence, which is really still part of the first wave of COVID-19. Since May, the end of May, our cases are starting to come down. Our deaths from COVID-19 are starting to reduce on a daily basis. There is good news temporarily. Don't think this is a moment to rest. However, everyone stay safe, continue practicing social distancing, wearing a mask, and being as safe as possible in social situations, because that will keep people safe moving into the fall. Now, the big story that's really interesting this week is about bradykinin. Brady who? No, not a Brady who. Bradykinin is a protein. That's right. Bradykinin is a protein that is in a pathway that is regulated by something called RAS, the renin-angiotensin system. And this is involved in blood pressure regulation and in fluid balance. And when a researcher at Oak Ridge National Laboratory was looking at the genes involved that are getting overexpressed in COVID-19 patients, he was looking through this and found that the genes related to RAS were way out of line. They were not doing what they normally do. And so followed this trail, and the study published in eLife suggests a new pathway that researchers can be looking at for treating COVID-19, suggesting that the RAS pathway and bradykinin are highly involved in the inflammation process that occurs. So the the RAS system, they followed this RAS, renin-angiotensin system, was abnormal in the lung fluid samples, led to what's called the kinin cascade, which is an inflammatory pathway, and it's regulated by RAS. So RAS goes up or down, the kinin cascade goes up or down. This kinin cascade is regulated by bradykinin. Bradykinin, if there's too much of it, causes blood vessels to leak. And when blood vessels leak, that means that fluid gets into the tissues where it's not supposed to be, and that leads to inflammation. So the fluid balance is off and inflammation takes place. Remember where COVID-19 patients tend to end up with a lot of fluid, Blair? In
Pneumonia?
their lungs. Yes, in their lungs, and they end up with pneumonia. This is part of this whole system. So the bradykinin, it makes the blood vessels leaky. Fluid leaks out into the lung tissues. And then there are other proteins that are there that interact with that fluid and create something of like a jello-like material. It gels up.
No.
Yeah, so the fluid, it becomes like trying to breathe through jello.
Ew.
Which is part of the problem for the oxygen transfer. It doesn't happen as easily because the fluid's there's too much of it, and it's thick. And so they are thinking that this bradykinin cascade, this kinin system, the bradykinin involved, can be a target, and other enzymes that are in there can be targets to really fix the inflammation and potentially target the pneumonia and other symptoms that occur. It's very interesting as a hypothesis, and researchers are definitely looking into it, especially because the renin-angiotensin system already has a lot of drugs that are in use for things like heart disease and kidney function. The renin-angiotensin system is already something that we have drugs that we're using that are already approved by the FDA that could potentially be repurposed. So this is definitely an interesting direction that it could go. So we now have many targets. We've got the ACE2 receptor. We've also got the ACE protein that's involved. ACE is also involved in targeting bradykinin. So that is a target. The RAS kinin cascade get in there. Hopefully we can fix it. This is promising. And I want to say thank you we sometimes talk about data mining of these big, giant genetic studies where they're just looking at all sorts of genes to try and find something that looks out of whack. Well, every once in a while, this kind of data mining really does pull something valuable out of the data. And this is one of those situations, which is very exciting.
So I just have to ask, because I guess maybe it doesn't even matter, but we were talking for so long about this being a respiratory disease. And then there was that big story, however long ago now, time is meaningless, where they were like, no, it's a blood disease. So
yes, that was a story. Yeah. Yes.
So would this,
and this would
explain that. This would explain how it's a blood disease, right?
How that part of it for sure. Yeah. It would explain how there is the leaky blood vessels, would also explain how there's potentially influences on the brain. That would explain why wasn't there like
COVID foot was a thing too, and that had to do with circulation?
Yes, it has to do with circulation. This would definitely explain those kinds of symptoms. Yeah.
Okay. We're just peeling away the onion on this thing. There's so much going on. Wow.
Yeah, but it's one of those things where at this point with medicine, it's wonderful. We're able to really dig in, and we've got so many resources targeted at this field right now at finding out, unpeeling the onion of COVID. We've got everything targeting it right now. And it's amazing how much we are learning in such a short period of time. I don't think we have ever seen this rapid amount of discovery on diseases before.
Yeah, that's a very fair point. And when we've had pandemics in the past, there's been a little bit of flying blind where you kind of try, you say, okay, so based on this evidence, I'm going to try this thing. But if we really can figure out exactly what is causing symptoms, that can help us treat symptoms, but it could also help us kind of attack the thing at the source in a way that maybe we couldn't have before.
Hey, Justin, did you have a COVID-19 story that you wanted to talk about?
I just got a quick blurby thing.
Yeah.
This was a meta-analysis, which is also a thing that we haven't really had access to in many past diseases throughout all of human history. They looked at 7 different randomized trials that included 1,703 patients, of whom 647 died. Well, that's about a third of them died. This is also these are very severe cases, patients who weren't doing well to begin with. They did 28-day all-cause mortality was lower amongst patients who received corticosteroids compared to those who received usual care, placebo, or some other treatments. So basically, it's the idea is there's effective therapies for patients with coronavirus who have, according to this, have demonstrated that low-dose dexamethasone reduced mortality in hospitalized patients.
Dexamethasone.
Yes, who required, with COVID-19, who required respiratory support. The interesting thing about this study, too, is just that it's a metadata study. There's just one of these things that we've sort of talked about, like, why isn't there more of this? How come we can't just look at the raw data throughout all these millions of cases that are going on to suss out what is and what isn't happening, what isn't working. Anyway, here's one example, and what did you call it? How do you pronounce the drug?
Dexamethasone.
Dexamethasone.
Yeah, it's a steroid. Yeah.
It's one that is rather common, used for all sorts of different applications in treating patients. So it's something that we do not need to guess side effect. We do not have to guess health implications. It's well documented already and is showing an efficacy at that late stage for the most severe patients.
Oh my goodness. This is This Week in Science, everyone. Do you want to help TWIS grow? Get a friend to subscribe today, please. That would be great. And do you know what time it is right now?
What time is it?
It's time. We are switching things up a bit this episode. It's time for Blair's Animal Corner.
She loves our creature, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, I have a story about anemones. And their fronds.
I love saying that. Anemone.
With fronds like these, who needs anemones? No, it's about their arms, actually. So this is a study looking at what determines how many arms an anemone has. So anemones, they're cnidarians. They're related to jellyfish. You can kind of picture them as a sessile. It means they don't move. Upside down jellyfish. Pretty much. So they have arms just like a jellyfish does. It's how they catch their food. And they all seem to have a very different number of these arms. So in mammals, our genetics tell us how many arms or legs we might have. We don't have any external kind of external variables for the most part that can alter that in development. But anemones are different. So until now, we haven't really known how many arms or tentacles the anemone can grow. But now this study coming from the Gibson lab at the Stowers Institute for Medical Research in Kansas City has shown that the number of tentacles is defined by the amount of food they consume. So, quote, controlling the number of tentacle arms by food intake makes the sea anemone behave more like a plant, developing new branches than an animal growing a new limb. So, reminder, they are animals. They're invertebrates, just like jellyfish, like I said. So, they are not plants. They are animals, but they do in fact seem to grow more arms if they are fed more. So this happens not only when they're a juvenile, but also throughout their life, which is helpful because if you lose an arm, for example, if a fish tries to eat one of your little tentacles, you can grow one back. And so it might actually be pretty handy if you could activate that by eating more food to replace your damaged limbs.
I lost my arm. I'm just going to have a burrito.
Yeah. Yes.
Wow.
So sea anemones show us that it is possible that nutrients are not converted into excess fat storage. They can actually transform them into a new body structure.
I think it's amazing. I mean, we know that there are limb regenerative abilities for so many animals, but humans have a hard time with it. Although we grow our fingernails, our noses never stop growing. Our ears never stop growing. What? If we break a bone,
my nose is big enough as it is. Oh,
it's gonna keep going, man. Our ears keep growing, right? Our bones, our skin repair themselves. There are regenerative aspects. Our livers love to regenerate. If we lose a chunk of liver, it'll just grow right back. It's really great. But why can't we? And how could we make this regeneration possible? So it's fascinating to see in what we think of as an animal, a simple animal still. Yeah, this isn't a plant. Even though it's acting like one, it's not a plant, and it's just regenerating limbs.
And those tentacles are complicated structures. They're covered in cnidae, which is why they're called cnidarians. It's the little fish hooks, little stinging cells that they have that allows them to sting fish that get too close and try to.
So they've got multiple cell types that are involved. It's not just, I'm just going to take this one skin cell and just keep growing skin. Yeah.
Pretty complicated structure with muscles and these stinging cells. Yeah. So it's
someday I just want to eat that burrito. And
yes. Oh my goodness. Yeah. Like,
I wonder
I hurt my leg. Cut it off. I'll have a big meal. It'll be fine.
So obviously,
don't have a cow, or maybe do.
Obviously, a small sacrifice to make to regrow a limb. But I suppose you'd have to give up like coffee and alcohol. Sort of as if you were pregnant while you were regenerating.
Probably.
Oh, perhaps, yeah.
Maybe.
You'd have to sort of go on like the same sort of nutrient rich diet that a pregnant person, I'll say pregnant woman might have to.
Yeah, I think it would be a very interesting study to take a bunch of anemones and then chop off a couple tentacles and then pour alcohol or something else or other pollutants into different containers and see if it affects their tentacle growth.
You are both cruel and curious. I like it.
Indeed. As researchers sometimes must be.
Yeah.
Which is a great lead into my next story.
Oh, jeez.
Let's get it.
It's about ensuring better science through better care for lab mice, which is a topic we talk about all the time. I think it's going to be a big deal in the future because it makes nothing but sense. A happy, healthy test subject will perform more reliably. So this is looking at mental stimulation in mice and their environments that they are held in leading up to experimentation and during experimentation. So this is a study from the German Center for Neurodegenerative Diseases, DZNE, in Dresden. And they looked at the behavioral differences from different mice in different kind of living arrangements. So they designed a sprawling cage that made up of 70 interconnected cages arranged on 7 levels. So they had quite a little mansion for these mice. This was considered an enriched environment. So it had equipment designed to inspire play. It had plastic toys, it had tunnels, it had hideouts, and they replaced and rearranged this stuff every week. And so that's something actually that when you look at animals in zoos or aquariums, they're expected to be enriched many times a week because that's supposed to be part of good mental health for animals in captivity. So they enriched all these animals. They had this huge, amazing play structure / mansion. And then, so what they did is, to be very scientific, they took inbred twins so that they had no genetic variation. This is going to come into play in a second. They wanted no genetic variation in their cohorts. So they took 5-week-old mice. They housed 40 of these individuals in enriched environments for 6 months, and 40 of them in standard cages for 6 months. And then 40 spent 3 months in enriched cages and then moved to standard cages. So they have 3 different cohorts. So one is all enriched, all bored, or enriched for a while and then bored. And then they were microchipped to record their movements. And then later they studied their brains, which did in fact mean they had to euthanize them to get to the brains. Thank you, lab mice, for your service. Lab mice that spent the entire 6 months in the enriched environment were more likely to explore as adults. There was a great range of individual personalities. And so they started to develop kind of their own way of moving around space and responding to test behaviors. In the context of mice, when we talk about individual personalities, that's relatively stable differences in behavior. So basically, it got to the point where you could kind of predict what this particular mouse would do. That would be part of their personality. Then they found that the mice that spent either 3 or 6 months in the enriched setting had more neurons in their hippocampus, which is the part of the brain associated with memories, learning, and emotion. The group that went from enriched to standard settings continue to show high rates of exploration, even though some of their activities are diminished. They still seem pretty interested in their environment. And they also found that the mice that had been kept in enriched environments had major changes to their genomes.
Mm-hmm.
Their genome changed from the space they were living in.
Wow.
Yeah, so their behavioral differences became imprinted on their genomes, and it remained them even when they went back into their standard cages. So this is long-lasting differences in their brains. They had methyl groups attached to particular regions of the DNA in the neurons of the hippocampus, and that alters the genome, and it alters how it's read and indicated. Mm-hmm. Exactly, yeah. So it changes the proteins that are made, basically. Yeah, so it's definitely an impactful change in their genome. And so on one side, this is really good news, potentially for people who have had traumatic events in adulthood, but whose childhood was otherwise stable, because it appears to show that when they're developing or where you spend the majority of your life can have an impact on your genome. But they think that because the mice that went from an enriched environment to their stable environment, they called it, continued to have this genetic change throughout the study. It was a short study, but still, it appeared to be unaffected. So one single event, kind of is what they're implying here, is not enough to mess up your genome forever. And that providing a good environment early on can have a permanent impact. So they didn't study old mice versus new mice in these enriched habits, but the implication is that where you grow up is pretty important, which I'm sure we all know. But I think it's more interesting because this does impact behavioral studies. If you're using mice in behavioral studies and they're being kept in a really boring drawer in a lab, that could impact your study.
So I've told this story before, but I was a long time ago in high school. I took a summer sort of internship job at the orthopedics research lab at UC Davis, and was headed by Neil Sharkey, who was the head of that department. Who went on to be head of R&D at Penn State, and I think has since retired. But I'd gotten this desk next to a bunch of mouse cages. And they were experimenting with this new needle. They would cut the jugular vein of the neck of the mouse and suture it up under this really intense microscope. And the survival rate was about 18% in these mice. And they just sort of hung out at the bottom of their cage. They were post-surgery mice that were stationed next to me. And their food was in the little, in pellets at the very top of the cage. And they, these recovering mice never seemed to go up to it. So I was sitting next to them, innocently enough, would feed them parts of my lunch. And I just sort of put it into the cage. They could get that, a little bit of cracker, a little bit of tuna sandwich, or whatever I was having that day. And they sort of, then they got just enough food. I didn't know what I was doing. All right?
You're not supposed to feed them snacks.
Well, after that, 100% survived. Because they were able to get nutrients that they needed without climbing to the top of the cage. And they sort of recovered and were managed to get up to the top of the cage then and eat the pellets. And it was just such a dramatic turnaround. Turns out, though, the experiment wasn't about survival rate or anything like this. It was just practicing with this needle.
Oh.
So 100% of the mice were destroyed anyway. And I didn't ruin somebody's experiment.
Yeah.
But it did prove to me that, absolutely. How you interact
with the animals
makes a huge difference to the outcome of the animals.
Yeah. And just to piggyback on that, there was another story that came out this week from Newcastle University, just showing that how you handle the mice can have a huge impact on their mood and how they respond to different stimuli. So they transported some by picking them up by the tail, which stresses them out, and they transported some by tunnel handling. So basically they encourage them to go into something like a paper towel tube and put a hand on either end and then take it to the next thing. Something just very simple like that. And so that one does not stress them out. And the difference is that the anxious, depressed mice, the ones who are being picked up by the tail all the time, are more disappointed when something bad happens, but their low mood has no effect on how elated they are when something good happens. But so basically, it does impact their behavior. It impacts their mood. And so we should be tunnel handling mice, which makes perfect sense. It might take a little longer, but it is totally worth it because it won't impact them negatively. And I'm seeing a lot of questions in the chat about laboratory animal euthanasia and all this kind of stuff. And there's some speculation going on. And I will say, as far as I know, the grand majority of lab rodents are euthanized via carbon dioxide gas, which is because it's a natural gas that's already in their body. It doesn't impact research. And they just go to sleep. And then everything that has to happen happens after that. I will also say that some labs donate their animals that were not used in chemical trials, that were used in behavioral trials, to zoos and wildlife centers to provide food. So they recycle, which is cool. But
there are many ways. Yeah. Something that I think is really, we've talked for years about how animals are kept, and we've talked about enrichment, and we've talked about, it was years ago that the hippocampus was shown to have neurogenesis in response to environmental enrichment. So I think this next step is fascinating because it digs even deeper into what's actually happening at the level of the neuron, what's happening in the cells. And so if at the level of DNA, you have these epigenetic changes, these are mice, but like you said, Blair, the implication that changing one's situation in the laboratory or in life can have long-lasting effects after that situation. So,
which is important in the pandemic, because if you have been spending since March inside and not enriching yourself, that could have long-lasting impacts on you. So go outside, take a walk.
Or also
get a giant wheel and put it in your living room.
Yes, it can change. So it's
the plasticity,
not set in stone.
It's not set in stone. It means that if you feel like your neurons are going, I can't handle this, and you're mentally not happy, after the pandemic is over, you're not going to stay unhappy forever. This will change. Yes. And your brain is plastic and it will come back. And these little tags that that get tagged in your neurons. I think the positive side of it is change can happen. And that's very exciting.
Yeah. And that
How can we
self-care
make
is
it
a
happen?
real thing. You have to enrich yourself.
Enrichment is great.
Yeah.
Is that your last Animal Corner story?
Mm-hmm.
All right. Everyone, thank you for listening to This Week in Science. Thank you for supporting us as well. You are the reason that we're able to do what we do. You're the reason we can continue bringing scientific information to everyone on a weekly basis, and we can do even more with your help. So if you are not already supporting us, please head over to twis.org, TWIS.org, and click on our Patreon link. Become a patron of TWIS. Select your level of support. Anything $10 and more, we will thank you by name at the end of the show. There are many levels, many rewards, but regardless, any amount helps us do what we do. So thank you. We cannot do this without you. Thank you for supporting TWIS. All right, we're coming back. Justin, do you want to tell some stories?
I've got one left. Our gut microbiota has been shown we've talked about this quite a bit on the show. It can influence our behavior. It can influence neurodevelopment. The intestinal microbiome composition has been linked to everything from depression, anxiety, autism, as well as neurodegenerative disorders, including Parkinson's, Alzheimer's disease, through metabolites produced by bacteria living in our gut. But what about our four-legged friends? And I don't mean cats, because they're not our friends. So it's just dogs.
Justin.
It's just dogs. Do dogs have similar responses in their intestinal makeup? Surely not. Since dogs can drink out of puddles, they eat anything, poop. They can do that, and they don't really seem to get sick. Or maybe more so because of all the weird things that dogs will eat and drink.
Well, I know that our microbiome is more similar to our own dogs than dogs' microbiomes are to each other, I think. Right? Isn't that a thing?
So, okay, good question. I know that there's going to be overlap between a dog and their owner. I don't know if that makes them more similar to the human than they would be to another dog, but there is an area of overlap in shared microbiome. The longer you are living with your dog, the closer your microbiomes sort of get closer to each other, but I don't know that it would make it
because you lick each other. I mean, you like each other. Yes.
But I have a feeling the dog carnivore gut from another dog is more similar to from dog to dog than it would still be from one dog to owner. But that's it. So this is new research, though, out of the anthology department, the Faculty of Science at Sloran University. Indicates that dogs' aging mechanism and memory performance are linked with their gut microbiome composition. So this is sort of interesting. They used service dogs for this because these are dogs that they can track over a long period of time. They have a wide range of lifespans, inclination to develop dementia, and environment shared with humans. So these companion dogs, promising model for the organism, for the aging research to be done. And it turns out it's pretty similar. Next generation DNA sequencing technologies have enabled the identification of the taxonomic composition and also the potential functions of the microorganisms, gaining a better understanding of microbial host interactions, says Thomas Fielold, assistant professor of the Department of Microbiology, ELTE, Budapest, who usually studies the microbial communities in natural waters. Sort of going into the microbiome now. Overall, what was sort of interesting is they saw two things. One was very similar. They saw there's a link in a particular gut microbe that is associated with Alzheimer's that also goes up in dogs as they age and as they start to do more poorly at sort of cognitive dog tests. It goes up in dogs as it does in humans. This is but there's some that do the opposite. They have things that head the other. Okay, so they found a negative correlation between the abundance of Fusobacteria phylum and the chronological age in dogs. Interestingly, in humans, Fusobacteria were shown to increase with aging, and elevated abundance of these microbes have been linked to serious illnesses like inflammatory bowel disease, colorectal cancer. So finding that the opposite is taking place in dogs was sort of an interesting find. My favorite part of this, though, this is Sarah Sander, who's a geneticist and apparently a Ziploc bag aficionado, who explains some of how they performed their science. After we tested the memory performance of the dogs at the Department of Ethnology, we took them for a walk and collected fecal samples. We had to immediately freeze the excrement in storage containers to ensure they would provide a valid picture about the bacteria that lived in the dog's guts before defecation. The time limit is important, as some species of bacteria can continue proliferation after defecation and therefore may falsely outnumber other bacteria in the sample. So they had to grab it and probably put it on ice, rush it into the freezer, and stop things from growing really, really quickly, which is not easy unless you get really good at get the system down.
Running all over the place, putting things in freezers. This is fascinating. I think it's it's fascinating that it's similar between humans and dogs, that these species go up and down.
The actinobacteria was the one that increases in both humans and in dogs and seems to affect short-term memory. It's one that's actually linked to Alzheimer's. Yeah. But the Fusobacteria was the one that went the other direction. It's the one that when it goes up in humans, we start to have all these bowel problems and stuff. And it seems like as dogs age, it actually goes down. So it may be a protective bacteria in that case. So it shows that the universality of a gut microbiome may not be there. That they may play very different roles depending on the species that they inhabit and have different affects. So.
Which would make sense, although we do like to think of, it's like they're mammals, they're animals like us. Wouldn't it happen similarly? Can't we just assume it's all going to work the same? Not necessarily. Not necessarily.
Justin, did you mention the sample size of this study?
No.
It's 29 dogs. So that's one of the things that because I was looking at this story too. I think it's really interesting, but it is a really preliminary study because it's only 29 dogs. So it's a very interesting look. I think it definitely means we have to look closer at dogs, especially since their microbiome is closer to us than pig microbiome or mice microbiomes are to ours. So studying the dog microbiome definitely has a lot to tell us about our own, definitely more than other common test subjects in laboratory and clinical trials and things. So I think more studies are needed, but yeah, definitely 29 dogs is a modest sample size.
Yeah. The other thing too is I'm now taking Dr. Justin's dog poo pills. Not a real doctor, maybe not poo from a dog. Pills off the market temporarily until we get further research done on this. Because
I won't ship you Sadie's then, like we discussed before. I'll hold off on that shipment.
Well, it never occurred to me until doing this study that there could be an interest in getting gut microbes from animals. Like A dog can drink out of a puddle and never get sick.
Mhm.
I want a dog's microbiome so I can drink out of a puddle and never get sick. Like, like people can write. Like, I never thought of that. Like, people could actually start requesting other species' gut microbiome to populate. That might not be good. Although, maybe there's a way not to be gluten intolerant with the right microbiome. Maybe there's a way to finally digest red peppers for me, or maybe it's if you want to eat a lot of bamboo, you can go get a panda microbiome and stuff.
So I just pictured. Are you guys familiar with the show Wild Kratts? Taking kind of a dark turn.
Oh, jeez. Yeah.
Mm-hmm. Wild scats.
Yeah.
Yeah. But I think the other aspect of it is, if we're seeing these shifts in the microbes of the gut bacteria of dogs and they're going up or down with the same kind of memory changes, maybe we can look at that and see how we can adjust our own microbiome better to avoid deterioration of memory.
Yes.
Yes. And, poor old dogs with dementia. That's always sad. Let's fix that.
How much you want to bet?
I have more stories.
Oh, I was just going to say, I'm out of stories. But how much do you want to bet that that we have probiotics for dogs?
Before humans?
Before we have a really comprehensive one for humans.
Cure dementia in dogs. Humans, not yet.
Yeah.
Right. Well, yeah. I mean, it's the supplement market that might get out there, but in terms of FDA approval, that's going to take a little bit more time. And who's regulating this stuff anyway? I have some stories about what I've got. What do we talk? Okay, let's talk about brains, because we were talking about microbiome and aging in brains and mouse brains, and now I want to talk about your brain on music. Your brain on music. Yes. When your brain is on music and you want to snap along to the beat, you're not just hearing the beat, you are also incorporating that understanding of the beat into your neural system, and then having a synchronized waveform of the activation of the brain, that your brain in a way is going to have to get synchronized with that beat. And then if you want to produce some kind of a snap or a tap that goes along with the rhythm, then you have to produce a synchronized output that matches the original rhythm. It's really a lot more complicated than it seems. So researchers wanted to investigate this, and they looked into, they were looking into some auditory rhythms. They had a simple auditory rhythm, they had a medium auditory rhythm, and then they also had a more complex auditory rhythm. The simple one was just 1-to-1. So the beat was just, and it was just at a very basic, easy to follow tempo, frequency. And the researchers had volunteers who were all experienced musicians. There were no musical novices in this group of volunteers for the study. Had them listen to these beats and then tap to the beat with either a 1-to-1, a 1-to-2, or a 1-to-1, a 2-to-1, or a 3-to-2 beat rhythm. So let's see. This is the first. There we go. This is the first rhythm they listened to. Seemed pretty simple, right?
Sounds like what a dripping sink sounds like when you're trying to sleep. Oh, why is it so loud?
But you're thinking, I can keep up with this beat. 1, 1, 1, 1, 1-to-1 rhythm. Pretty easy, right? You can do that. And then the more moderate rhythm, Keeping up with one out of the two of those beats is a little harder because you're dropping a beat there, right? It's a little bit harder. Okay, and then the more complex rhythm. Hold on, here it comes. It's a little bit.
Chat room is really sure that we're about to get rickrolled here.
There's no rickrolling going on here. This paper was published in the Journal of Cognitive Neurosciences, and the researchers did actually identify markers in the brain that determine differences in the musician's ability to do different aspects of this task. And they found that the brains had an easier time synchronizing with the simpler rhythm than they did with the more complex rhythm, and that there was variation between individuals on how well they were able to synchronize with the rhythm, and also how easily their motor output synchronized then with the with what they originally listened to. But they found that the brains of these individuals, as the rhythm became more complex, yes, more of the brain was incorporated into synchronizing with the rhythm, not just listening to it, but actually figuring out the rhythm to be able to have a motor output.
The better than coffee for me to get my mind awake and active and get my whole body and mind moving and functioning, swing music. Yeah, there's this electro swing station I've been listening to. It's swing, but it's also like sort of modernized and redone. Those beats, for whatever reason, get my mind turned on and active better than anything else. And it makes total sense that there's some sort of synchronizing. My neurons are like, oh, a lot of stuff is going on right now. Let's jump in.
Yeah. They also found that the ability to hear the music, so just the auditory aspect of just listening, that had absolutely nothing to do with their brain's ability to synchronize with the music and to basically understand the rhythm.
No, you got to dance. You got to start dancing.
Yeah. So the listening, the stuff comes in, and then your brain does something with it, and then it has an output, and it's whatever your brain does with it. And some people, their musicians, are either better at synchronizing or worse. Does that make them better musicians? I don't know.
I think it depends on what kind of musician. So
right.
My personal experience, because I played the baritone saxophone, is I was often in charge of the bass line. And my favorite bands were always the jazz bands that I played in. And so I was in charge of this very like steady beat in the background when other people were flip-flopping all over the place, having their fun. And so I think that was something, that I had to be very careful. And I think I was pretty good at keeping that beat on and maintaining that while everybody else was doing their crazy thing. Maybe the drummer was having a drum solo, not maintaining a beat almost at all. So that was definitely, I think it takes different types of people to do different parts of music. And that's something that's really important if you're in charge of a bass line or you're a drummer.
What aspect of it? Yeah. What they did say, what this quote from the researchers I find really interesting. We were surprised that even highly trained musicians, that's highly trained, that doesn't necessarily mean they're good. Even highly trained musicians sometimes showed reduced ability to synchronize with complex rhythms. And this was reflected in their EEGs. Most musicians are good synchronizers. Nonetheless, this signal was sensitive enough to distinguish the good from the better, or super synchronizers, as we sometimes call them. So apparently there are some individuals, like super tasters, like super smellers, their brains are super synchronizing to a beat. So another brain story that happened this week, last Friday, Elon Musk announced, had another press conference for his
Oh, dear.
Neuralink technology. Yeah, and the
I don't know
Neuralink,
this one. I haven't followed this one.
this is one of his companies, and they are trying to develop brain implant technology that will someday be used to treat brain disorders. But more recently, he's really doubling down on creating a human ability to integrate with computers so that we don't get overrun by AI. Because if AI is going to be super smart, then we need to become super smart as well. And how do we do that?
Aren't
We
you
meld
just
with the
handing the
machines.
AI the keys then?
I feel like exactly. I feel like that is exactly what's happening. Like when you are identifying your friends and family for Facebook, you're feeding facial recognition. You're just doing the work for improving facial recognition when you do those sorts of things. When you identify things in those crypto picture-y whatchamacallums to get into what do they call it? When the visual ones are like, where's the street sign, or which one of these is a boat?
I guess, is that a CAPTCHA?
Captchas, thank you.
CAPTCHA?
Yeah, you're absolutely training artificial intelligence every time you do that. So yeah, letting the AI into the brain?
Yeah.
No, thank you.
Come on in, computers. Come in. It's okay. But are we really getting there? This week, on Friday, what he announced is his device. Last year, he announced a robot that was going to eventually do brain surgery to put a brain implant behind your ear. But now they've kind of changed their plan, and they are talking more about developing an interface technology where the robot would implant a little plug into the top of your skull, and the plug would contain about 1,000 electrodes, little tiny wires, someday hopefully up to 10,000 electrodes that would then interface with neurons in your brain to collect signals from your brain that could then be read by this device, which is Bluetooth enabled. And the Bluetooth enabling would allow the device to then talk to computers. The computer could then potentially do things. He showed in the press conference the device being used as it's currently implanted in a pig named Gertrude. The pig has an implant, and whenever her snout is touched, there's an array of electrical activity from neurons being activated in the brain that go bloop, 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 bloop. And so it showed a very musical display of neuronal activation. Is this an advancement on anything that we currently have? The answer so far is no. We already have brain implants. And in fact, there was a story from just in April this year of a flexible neural interface that was published in Science Translational Magazine, Science Translational Medicine, that has over 1,000 electrodes in this device that would would outlast and outwork the implant that Musk is talking about. So this device is not that Musk is selling at this point, has not yet been approved for human use. He's gotten breakthrough technology ability from the FDA, so he can start looking at it in people. But so far, it's just been animal trials. They're looking at human stuff in the future. Is it a big deal? So far, the verdict is no. That doesn't mean it won't be, because he is recruiting very intelligent, enabled engineers to be able to develop this technology further. Will it become the thing that lets us be equals with the AI? That is definitely yet to be seen. So I still need to talk about space and dark energy.
Let's go.
Because we're not done with that story about dark energy. There's a lot of it. Dark energy, right?
Huh?
A lot of dark energy in the universe. It's pulling us apart. Dark energy, it's causing things to spread out all over. You know, dark matter is the clumping. Dark energy is the expansion of the universe, which is accelerating It's like someday we're going to all be spread so far apart from other galaxies and other places in space, we won't be able to see them or detect them in any way. Where is the dark energy? How do we find it? It's dark. What is it? There has been a hypothesis for decades that not all big old stars form black holes, or at least not black holes in the way that they are described by physics, that there's an alternative kind of black hole. It's called a geode, generic object of dark energy.
Ooh.
Yes.
Does it have crystals inside still?
No, it has dark energy inside.
Oh.
And that when big old stars collapse, they don't just form a black hole, they don't break Einstein's equations. There's no singularity. What happens is you get a spinning outer layer around a core of dark energy. And it's dark, so we can't see it. And it acts just like a black hole, except it's not. It's the black hole's dark cousin that we don't talk about. Anyway, the black hole, and there's been some evidence in gravitational wave detection that some of the mergers of black holes that we're seeing, they're bigger than they should be. And so one of the explanations is that they're not really black holes that are merging, but they're actually these geodes that are merging, and that they're mergers of dark energy. But that's another side story to all of this. Researchers at the University of Hawaii in the Department of Physics and Astronomy, they've been working on this idea of geodes. And last year, they showed that geodes could, in principle, provide the necessary dark energy that's in our universe to explain dark energy in our universe. If they moved by like black holes, say, one of the researchers, Kevin Croker, staying close to visible matter, galaxies like our own Milky Way would have been disrupted. And obviously our galaxy hasn't been disrupted, so they're someplace else. And so they've just published in the Astrophysical Journal a new study looking at the dynamics of these geodes and their spinning. And they put the spinning of these geodes into a computer simulation and determined how they moved relative to each other. And they found that if the outer layers spin slowly, the geodes love to clump, and they actually clump more than black holes. They're super clumpy. But if they spin super fast, near the speed of light, then they avoid each other. They're like tops that don't want to be near each other. They're like, boom, let's go to the other edges of the galaxy. And they repel each other. What this means, they solved a bunch of equations and looked at energy and mass distribution in the universe, and pretty much the dark spaces in the clumpiness of our universe are probably filled with geodes, and that would explain everything. They think that the distribution of matter and dark energy in our universe is explained by the fact that these geodes are there. They're these big collapsed old stars that ended up being dark energy spinning around really, really fast and repelling themselves and everything else. And that instead of hanging out with places like the Milky Way and our black hole, they're like, I gotta get out of here. And they are leading to the expansion of our universe. I have a question from Jason Olds. Jason Olds writes in, I am a longtime listener and Patreon member to TWIS. When you started your new segment with listener questions, it made me think about what I may have a question about. I started to think about small things first, viruses, bacteria, amoeba, fungus, parasites. Then I came across enzymes. I went on YouTube and watched a quick video that was way over my head. I realized I know 0% about enzymes. Do you mind giving some basics on them and why they exist in our world? Well, Jason, let me tell you about enzymes. Enzymes exist to speed things up. Enzymes are what we call Cadillacs. Not Cadillacs. They are
Cadillac.
not a Cadillac. Enzymes are
They're
what
the
we
Cadillac
call
of proteins.
catalysts.
Thank you.
Catalysts. Yes, or what I like to call, they're like biology's power up. Beep, 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 beep. You can suddenly do things you couldn't do before. In fact, there have been enzymes discovered that speed up biological chemical reactions so much that they allow life to happen. In fact, back in 2008, there was an enzyme that was discovered, an enzyme that is called uroporphyrinogen let me try this over again. Uroporphyrinogen decarboxylase. I cannot speak tonight. I can't wait to do the Patreon names at the end. This decarboxylase, enzymes end with an A's in our terminology in biology. Uroporphyrinogen decarboxylase. It changes the rate at which chlorophyll and hemoglobin can be created. And if that enzyme were not there, it would take like 7 billion years for hemoglobin to be created. We would not have the molecule that can carry oxygen because it would have taken half of the... it would take a really long time. Anyway, yes. Chemical reactions take a very long time and the energy, there's this energy level that you need to get over, this hump, this threshold that needs to be gotten over to allow the reaction to take place. And so enzymes are this wonderful metabolic tool that cells, sometimes it's just a chemical, but sometimes they are molecules that connect together to allow the energy to be accessed more quickly. It's like a quick elevator lift. It's like, whoop, there you go. You made it to the top. Now you can go down. Or they reduce the energy of activation so it's not as hard to get into the reaction in the first place. Enzymes,
they're the proteins in your body doing all of the work. Yeah.
They do all the work. Without enzymes, we would not be able to do anything. They are targets for drugs. Targets for many, many drugs, because when you adjust how enzymes work, you adjust the rate of reactions. And so you can affect diseases from even happening or not. Yeah. Enzymes. I hope I answered your question.
You need enzymes to consume dairy. I always think about that. Very specific.
I do.
Enzyme
Lactase.
that you need. So
yes.
Thank you, lactase, for allowing me to eat cheese and ice cream.
You have salivary enzymes that pre-digest your food for you so that it's not as hard to chew and not as hard to digest when it gets in your tummy.
Good job.
Yes. Yes.
Good job. Good job, enzymes.
Thank you for digesting my food for me, enzymes.
And one way to visualize what an enzyme does in terms of work, in terms of being a catalyst, what that means as a catalyst is you can think of an enzyme as vinegar to your bowl of baking soda, right?
Mm-hmm.
Got the big bowl of baking soda, just sits there, nothing happening. The enzyme is your vinegar, you pour it in, suddenly everything is exploding. All this energy has been released. And all this chemical reaction is taking place at a really rapid pace. It's basically what an enzyme is doing. It's unlocking the energy and transforming those molecules. We use it for everything. Laundry detergent. All laundry detergent.
Laundry detergents are enzymes. Yeah.
They're all enzyme
very
based
important.
Yeah.
Enzymes are responsible for so many things. They are essential. Tenderize your meat, clean your dishes, allow you to live. That's right. That is right. Okay. We have come to the end of the show, and I just do want to say at the end of this show how much we are going to miss Ed moving forward. I found The earliest email I have from Ed in my Gmail, that's not even going back to my other old email accounts. Edward Dyer sent me an email. He started sending me TWIS stories for the show, and he sent them in emails. And he said, "You've probably seen this story, which gets my vote for one of the best science headlines of the year. Parasite causes zombie ants to die in an ideal spot. Hollywood could be kept busy for years just making movies about parasites that exist in the real world. You could do an entire podcast on parasites. So could someone who does political shows." Smiley face, Ed. Yeah. That story was from August 13th, 2009. We're going to miss you, Ed, very much. And if you are listening to the show and you know Ed, we are going to organize a Science Island gathering to be able to get together at some point soon so we can talk, share memories, and just be together and remember. So stay in touch, keep in touch, get in touch if you're interested in that, but I will be reaching out. Thank you all for listening. Shout outs to Fada for helping in the show notes and for helping with social media, making that happen. It's amazing. I really appreciate your help. Gord, thank you for manning the chat room. Your assistance there is essential, and Identity 4, totally essential. Thank you for recording the show. And I would like to thank our Patreon sponsors and the Burroughs Welcome Fund for their generous support. Thank you to Donathan Stiles, a.k.a. Don Stylo, John Scioli, Eric Combs, Flying Out, Guillaume, John Lee, Ben Rothig, Ali Coffin, Maddie Perrin, Gaurav Sharma, Josiah Zahner, Mike Shoemaker, Sarah Forfar, Donald Mundis, Rodney Lewis, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Corinne Benton, Sky Luke, Paul Ronovich, Ben Bignall, Kevin Reardon, Noodles Jack, Sarah Chavis, Paul, Jason Olds, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, Sue Doster, John McKee, Greg Riley, Mark Hessenflow, Jean Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brandon Minish, Melazon, Johnny Gridley, Richard Porter, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood, Robert, Rudy Garcia, Dave Wilkinson, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Alex Wilson, Dave Neighbor, Kosti Ranke, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Patrick Pecoraro, Gary S., Ed Dyer, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you all for your support on Patreon. And if you are interested in supporting us, you can find information at This Week in Science. Click the Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, broadcasting from our YouTube and Facebook channels and from twist.org/live.
Hey, do you want to listen to us just only with your ears? We're a podcast. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, hey, get a friend or a family member or an enemy or a coworker to subscribe to.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, which is www.twis.org, which is O-R-G. And you can also sign up for our newsletter, which is apparently a thing that...
Yeah. Hey, you got something to say to us? You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just put TWIS, TWIS, in that subject line, or what'll happen? It'll turn into a geode of dark energy in space.
Spin really fast, create a giant black hole, where you can also hit us up on the Twitter. We're at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you tonight, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science.
This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views. But I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought. And I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week in